So it's now time to board on Rover for true. Rover, which is the data visualization module of Cytonaut. This will be a vivid experience where you are the superhero biologist empowered to explore and decipher the single-cell universe. So here, once the post-processing run is done, you are propelled on a landing page where each point is a single cell projected in the 2D space with the color of the cluster it belongs to. Okay? So here, the goal is to identify the cell types of each of these colorful clusters. If you remember correctly, Diliana explained that to obtain such data visualization, you have played with some post-processing parameters. For example, the minimum transcript count per cell or the clustering resolution. For each set of parameters, there is a dedicated Rover page which will be generated. So you can iterate between interactive data visualization and parameter tuning in order to refine your interpretation until being able to perform manual annotation of the cell types with a high level of confidence. So this is what you will get at the end, and we will do that together on a typical example of PBMC application. First of all, I would like to give you a very concrete example of extreme value of parameters, so that you can better apprehend that word. First of all, to project the high-dimensional transcriptomic signatures of the cells in the 2D space, there is an algorithm called UMAP, which needs to know the minimal distance that you allow between 2 points in 2D. And that minimal distance is called min-dist parameter. By default, it is set to 0.5. But if you decrease it to the extreme value of 0.1, you will get a much more compact rendering where the local distances between the cells are much more preserved than the global distances. And the consequence is that you can see that some connections between cell components get lost. You see that? On the other side, if you increase that parameter to 0.99, you will see that this time the global distances are more preserved than the local ones, and this may create some artificial connections between the cell components. So you may wonder, what is the optimal value? Well, the optimal value is the one that gives you the best perception of the biologically meaningful structures. And the default one is a good one to start with, to be honest. Now there is another interesting parameter, which is completely orthogonal. His name is the resolution of the clustering. The resolution is controlling the granularity of the clustering. So the default value is 0.8. Now, if you lower it down to 0.1, you will see that some distinct cell components have been merged in the big orange here on the top right. And if you do so, you may lose some biological heterogeneity, right? If you do the contrary, you increase it to the extreme value of 3. This time you have too many clusters. It's called over-segmentation as opposed to under-segmentation. I would say that the over-segmentation is less a problem because you will always have the possibility to assign the same cell type to different clusters. So I would recommend to over-segment a bit rather than to under-segment for the next steps. So coming back to our mission, what we have done for you, we have pulled 2 replicate samples of PBMCs, technical replicates. And the first question is there a batch effect? So what is a batch effect? It's a sample-to-sample difference which can be caused by technical variability, not biological, but technical variability. To check that, you simply click on a cell attribute that we call sample name. And as a result, you will see the colors are different. This time, the cell is colored by the samples it belongs to. So either the teal color of the replicate 1 or the orange color of replicate 2. And we can see here how nicely the 2 color distribution are overlapping, which means good news, there is no batch effect in that mission. And in fact, with Cytonaut Rover, you can appreciate the 2D distribution of any cell attribute you want. So here we take the example of the number of transcripts per cells, but it can be the number of genes, it can be the number of mitochondrial transcripts, etc. So this is an example in logarithmic view, where you can appreciate how the number of transcripts are distributed among the cell types, which we don't know yet. So here comes the main goal of the mission. What is the cell type of each cluster now? Well, Cytonaut Rover proposes you two complementary strategies. The first one is a supervised method to confirm hypotheses, while the second strategy is a non-supervised method to discover information. And depending on which case you are, this can be very useful, and this can be also combined. Let's take strategy number one. Well, we are using PBMC samples. They are very well known, very well characterized in the literature. Let's take this paper from Monaco et al. Inside, there is a published gene list, so biomarkers for each cell type. We know which cell types are expected. We know the list of biomarkers per cell type. So let's copy-paste this list directly in Rover. So these are 14 genes. Immediately, we can see in each UMAP representation the gene expression of these biomarkers. Let's take a first question. Where are the natural killer cells, also called NK cells? I know that NKJ7 and JLNY are two typical biomarkers of natural killer cells. I know that because this is an a priori knowledge that I am using. So if I am looking at the violin plot, what we can see that both the cluster 4 and the cluster 6 are expressing both biomarkers. But are they really the same cell type? Maybe we need to disambiguize something here. I also know that there is a third gene which is called CD3D, which is not expressed in NK cells. It shall not be expressed. And here we see a difference. The cluster 6 only does not express CD3D. So this is a good match. Cluster 6 must be natural killer cells. To confirm this hypothesis, I can combine 3 filters. Be larger than 1, be larger than 4, and be lower than 1 for each of the genes. If I apply these 3 filters in Rover, I can highlight clearly the cluster number 6 at the bottom. And now it's time to annotate it with a high level of confidence. I grab my first star, and I say this is natural killer cells in cluster 6. Imagine we know nothing about PBMCs. It's difficult to imagine, but just play the game, okay? I know nothing. I don't have any a priori knowledge. So I will use strategy number 2, which is a non-supervised method. Let's look at these clusters, and I love this one, the purple one here, okay? At the bottom right. It is well isolated, and I would like to know what is the cell types behind this cluster. So with Rover, with a simple click on the ID of the cluster, ID number 2, I can access to the top 10 genes that are the most differentially expressed, positively expressed in cluster 2 when compared to the union of all the other clusters. So it's 1 versus all. And what do we see? We see that the very first one, MS4A1, is in fact a biomarker of B cells. This is well known. So we can deduce that cluster 2 is B cells, and we can now annotate it as well. This is our second star together. And so on. So you can use either one or another strategy or both, depending on the context of your applications. And at the end, you are very happy because you have annotated everything, and you can also access to the balloon plot of the biomarker signature of each cluster. And everything, as explained Delina, is exportable in image or vector format so that you can further customize them as you want for publications. We have done all of that together. We would like to capitalize on such a work. So that list of genes, it's possible to save it in order to reuse it for another mission. And you have also played with different views, right? You have selected genes, you have selected cell attributes, so you would like to save that as custom views that you can see later on. So you can save multiple views because, in fact, with your colleagues, you may have different viewpoints on the same mission. And in each of these views, the annotation you have made is also saved. And we may imagine different ways of annotating a mission, because we may want to be more or less granular in the way we do so. So maybe a colleague will do it differently. So it's time now to grab all your precious discovery and, as I explained to Elina, to share that with your colleagues. It can be a bioinformatician, it can be a biologist. In particular, the annotations you have made are exportable at cluster level, but also at cell level, so that you can directly import them to Geo. And it can also be useful for you, because it can become for you a reference data set that will allow you to go further in a next mission where you will look at much more detailed biological questions. And because you are using the same tool, Cytonaut, you are speaking now the same language. May you be a biologist or a bioinformatician. And this is very powerful, because this will enrich your discoveries. And the last thing to remember is that at CPO Bioscience, we have a dedicated support team, which is always here for you to accompany and to make you succeed in your single-cell missions.